Hello everyone, welcome to my channel. I'm Sharmin. Today I'm going to teach you lock and key and induced feed hypothesis. Okay, so let's get started. Enzymes are globular proteins. Like all globular proteins, enzyme molecules are coiled into a precise three-dimensional shape with hydrophilic R groups and side chains. That means on the outside of the molecule ensure that they are soluble. And enzymes molecule also have a special feature in that possess an active side. So you can see here the diagram. This one is an enzyme, this is a substrate, and this one is the active site. Okay, so the active site of an enzyme is a region, usually a cleft or depression to which another molecule can bind. Okay, so this molecule is the substrate of the enzyme, and the shape of the active site allows the substrate to fit perfectly. And the idea that our enzyme has a particularly shape into which the substrate fit exactly is known as the lock and key hypothesis. And the substrate is the key whose shape fits the lock and the lock of the enzyme. The substrate is held in place by temporary bonds which form between the substrate and some of the R groups of the enzyme's amino acids, and this combined structure is termed. Okay, so yeah, an enzyme may catalyze a reaction in which the substrate molecule is split into two more molecules as shown in here. Okay, so you can see alternatively it may catalyze the joining together of two molecules, as when making a dipeptide. A simplified diagram is shown in here, and the enzyme product a complex which is briefly formed before release of the product, and interaction between the R groups of enzyme and the atom of the substrate can break or encourage formation of bond is the substrate molecule forming one, two or more products. Okay, so yeah, and when the reaction is complete, the product or products leave the active site, and the enzyme is unchanged by this process. So it's now available to receive another substrate molecule, the rate of which a substrate molecule can bind to the enzymes and active site and be formed into products and leave can be very rapid. The enzyme catalase, for example, can bind with hydrogen peroxide, and hydrogen peroxide molecules split then into water and oxygen and release these products at a rate of 10 million per second. Okay, so guys, the understanding is so far. Okay, so I am here to help you, don't worry. The interaction between the substrate and the active site, including the slight change in shape of the enzyme and which results from the binding of the substrate, is clearly shown by the enzyme lysozyme. And lysozyme is a naturally defense against bacteria that is found in tear, saliva and other secretions, and it breaks the polysaccharides and polysaccharide chains that form the cell walls of bacteria. Okay, so yeah, and the tertiary substrate the tertiary structure of the enzyme has already been shown in here, so you can see here. Okay, so guys, that's all. Thank you very much.